Chapter 3, Section 3, Working with Proteins. Now, as I stated in a previous lecture in this chapter, well, you may want to study one protein in a cell. The problem is that's one protein amongst anywhere from 100 to 1,000 of different other proteins, and each of those proteins has anywhere from 1 to maybe 1,000,000 copies of it in the cell. So it's like trying to study one tree in the middle of a national forest. It's going to be difficult. So you have to have a way to separate out that one tree, that one protein you want to look at, you want to study. Well, over the decades, we've developed certain methods, certain methodologies for doing this. We can separate out a protein or a set of proteins based on size, overall charge, binding properties, what will it interact with, and its solubilities. To purify, first you got to get what's referred to as a crude extract. Basically, you need to take the cells that have the protein in them you want to study, and you go through and you tear them up. You break open those cells. You will get a soup of different proteins and all the other components of that cell that were in, that were in the cell are now in this mix. So it's called a crude extract. You can separate out based on who's soluble in whatever buffer you're using that you were using when you isolated or you broke open those cells. Next, you're going to go through and you're going to do what's referred to as a fractionation. You're going to separate the proteins into fractions, into little subsets based on many different methodologies. A good one is what's referred to as salting out. You make a protein salt. You precipitate it out, and it associates with different salts that are now in your fractionation buffer, and it will settle out based on centrifugation speeds, based on many other things. So you get this precipitate of proteins, not just the one you're interested in, but many, many others. But they're now in a salt. They have a lot of salts that are now interacting with the R groups, all those ionic R, those ionic R groups in those amino acids. Then you have to go through and do what's referred to as dialysis. You take your fractionations, you put them in a dialysis tubing, in a dialysis bag, and you put that in a much, much larger volume. And you allow osmosis and diffusion to pull the salts out of, remove the salts from your protein. And you go through and you do many, many steps of that. So hopefully you now have a purified, or at least enriched, protein. There's other ways to do it. You could do what's referred to as column chromatography. Hopefully you remember doing this in 244, in Biology 244. You go through and you take your cell, your crude extract or whatever, and you run it through a column. This column is going to be filled with some sort of solid that's either going to be microbeads or it's going to be some sort of cellulose thing that's going to interact with certain proteins. Not all of them, only certain ones, and hopefully one of those that will interact will be your protein of choice. So as you notice above my head here, as your big mix, your crude extract or your fractionation, whatever, goes through, things start to separate out by size, by ionic interaction with the agar, or the microbeads or the cellulose that's been treated a special way. And things will slow down and will begin to separate out. As you notice there, ABC will separate out. Some things, all the stuff you don't want, just flows through. That's the waste. Now you just have to come up with ways to change the buffer that's running through there and allow whatever A is above there in the column to release. Change the buffer again, B will now release. Change the buffer again, C releases, or you do versions of that. Now, are you always going to know when A comes out, when B comes out, when C comes out of the column? No, because it's not going to be here like it shows here when everything's colored. Okay, it's all clear liquids, all clear solutions. So you just have to keep taking samples in little tubes. Here's 20 drops, here's 20 drops, here's 20, here's 20, or 10, 10, 10, however many you want to do. And then you go through and test. Tubes 1 through 5, nothing of interest. Tubes 5 through 10, well, this seems to be where C is. So you pool all of those together. Tubes 10 through 18, well, that's where seems to be where B is. Pool those together, and you just keep going and going. Again, is it going to give you completely purified proteins? Well, no. But you've enriched it till the majority of what's there is going to be your protein of interest. There may be a few other proteins that have, come along for the ride, but you can either, repeat this again and again, or you can go to other methods to further attempt a purification. Ion exchange here is great. I like this one. Here, the column one, a lot of times that's going to be size. Here, ion exchange, literally, you got to remember proteins at different pHs are going to have overall charges that are going to be different. So, a resin, microbeads, that's overall negative charge, then proteins that have an overall positive charge will adhere. Everything else flows through. Change the pH. Your thing of interest now, will go through. And again, you collect the fractions. You collect the tubes, multiple tubes. Then you test them and pool the ones together that have your compound, your polypeptide, your protein of interest. Size exclusion is another one that's going to be good. That it's going to interfere or allow certain things to pass through based on size. Depending upon which gel you're using, it could slow down big things, only allow little things through. Or it could go the other way. Little things get ensnared, only big things get to go through. How do you know what's going to happen? Well, it turns out there's dozens and dozens of different types of gels, different types of substrate that you would put in here for the things to run through. Do I want to exclude big? Do I want to exclude little? Do I want to exclude both and only get the things in the middle in this very narrow band of size? Well, there's a gel for that to be used in the column. You just have to know. You have to go through and do some research on which ones you want to use and which pressures, which different wash solutions, which different buffers you're going to need, and so on. Affinity chromatography is good because proteins, as long as they're in their native state, are going to want to interact with other proteins, or there's going to be some sort of a compound or some sort of organic molecule the proteins can interact with. Well, if there's a certain protein, a certain polypeptide, oligopeptide, or some form of a molecule that your protein of interest will interact with, well, you just coat these microbeads with it. And then as you put that extract through, your proteins of interest will adhere to the microbeads. Everything else just flushes through. You come in with, a slightly different buffer, and then you basically, run that through the column, and your protein of interest will release from whatever it's bound to on those microbeads. Again, you don't know exactly when it's going to come through, so you collect the different fractions, test each fraction for, the protein, which ones have the greatest concentration, and you pool those together. Nowadays, instead of letting gravity, like if you did this back in BSc 244, an intro cell and molecular, you just put it in the top and let it drip out the bottom, and that was gravity driven, the weight of the liquid pushed through. Well, that takes time, okay? Especially you have to go through a large volume of extract, and you really want to be precise. Well, nowadays they have what's referred to as high performance liquid chromatography. HPLC is the acronym for it. This will use pumps, the same gels, the same microbeads and all of that, the same buffers, the same wash buffers, same release buffers, all those stuff. But now it's using pumps and high pressure. So instead of taking anywhere from 20 minutes to an hour, like it would if you did it the old-fashioned way, using water pressure, just using gravity and water, the liquid, weight of the liquid to push through. Now you're doing it under pressure, and it can do all of that within minutes. And because it's doing this, and you can go through and you can run multiple washes through to remove the extraneous stuff that may have just gotten caught up. And you can still do it within the minutes as opposed to the half an hour to an hour the old way used to do. And that's without all the washes and all the other things. So when you approach this as a biochemist, and I wish we could do this in lab, but we didn't have enough time to set this up. You would go through and you would take, as you see in step 1, a crude extract. That's basically you just busted open the cells and you collected that soup. Look at the volume, 1,400 milliliters, 1.4 liters, 10,000 milligrams of protein, and that activity units, our enzyme of choice. When we test that crude extract, we have a scale, sliding scale of just how active that enzyme is. The more active it is, we say, the more of that enzyme has to be there. Okay, so 1.4 mLs or 1.4 liters, 10,000 milligrams of protein, 100,000 units. So that's about a specific activity of 10. Okay, it's a relative scale, 10. You go through and you precipitate with ammonium sulfate. Remember, do that salt precipitation. The ammonium sulfate will form a salt, a protein salt that under centrifugation will settle out. So you've gone from 1.4 liters to 280 mLs, or 0.28 liters. We've lost about 2/3, a little over 2/3 of our total protein, because not all the proteins are going to settle out. Active units, gone from 100 down to 96, not a problem. But we've increased our specific activity, our scale. We've gone from 10 to 32. Do the ion exchange. Okay, gonna go through, clear out some more. Go from 280 mLs down to 90 mLs. Okay, so we're about a third. We've lost 2/3 of our total volume, because remember, we're doing all the fractions, pooling only the fractions of interest. Started out 10,000 milligrams of protein, we're now down to 400 milligrams. We've gotten rid of a lot of that extraneous. 80,000 units, we're now to a ratio, we're now up to 200 units per milligram. We've started at 10, we're now 20-fold higher, 200. Use the size exclusion. Okay, get rid of things that are the wrong size, too big, too small. We want the Goldilocks, just right. Go from 90 mLs to 80. Okay, 400 milligrams of protein down to 100. 80 to 60,000 units. But we've now tripled our specific activity, which means that the more of that protein is there, the more of that protein that's present that we have in our tube is our enzyme of choice. So it's not pure yet, but it's getting there. Okay, we're going through steps, we're enriching. So instead of being one of thousands at the beginning, it's now one of hundreds. Maybe one of dozens of proteins there. Finally, we do the affinity chromatography. It turns out our protein of interest, our enzyme of interest, has an affinity. It can bind to something, a metal. It can bind to, biotin or something like that. look at our volume. We went from 80 down to 6 mLs. Our fractions, we've collected them. We now have 6 mLs. We started at 10,000 milligrams of total protein. We're now down at 3 milligrams. Our activity unit started at 100,000. We're now down to 45,000. Our specific activity, 15,000. Started at 10. We've now gone up 150 or 1,500 fold. Hey, do we have a completely purified protein? Probably not. But now, instead of being the one of thousands that you started with, it's now probably one of just a handful. But it's the protein that's most represented there. That's something we can start studying. We can also utilize what's referred to as electrophoresis. Electrophoresis, you're using an electrical field to separate proteins based on their overall charge. Then we can take it a step further, because we can have, instead of having these proteins free floating and just going towards the cation or the anionic, pole, we put them in an agarose gel. So now we're impeding not only their movement, we're making them move, but we're impeding their movement based on size. And it gives us a way to, smaller things will move farther, bigger things, bigger proteins are going to impede it to the point where they won't move much. With this, you see here just above my head, up here is the start. Oops, sorry, that's a really bad S. Up here is the start. Everything started up here at the top. And as you see here, everything's moving this way. Start up here. There was a well. You put your sample in. And we've pre-treated all of these proteins in such a way that they have one overall charge, okay? An overall negative charge. So they're going to move away from the cation towards the anion, away from the negative towards the positive pole. Up here at the top, these are the big things. They didn't move very far, because right here is going to be the well. They don't move very far. Small things, even smaller bands you can't see down here that went off because they were so small. Now, why can we see this? Well, because everything was stained with Comassi blue dye. Comassi blue has an affinity for amino acids, amino acid chains, polypeptides. So in the gel, this was all clear. Take the gel, you put it in the Comassi blue stain for a while, you rinse, add in some rinsing instead to remove any excess Comassi blue that's not stuck to proteins, and voila. There you go. You see here in this, they were trying to express proteins. What did they do? Here's the induced cells. You notice that there's a lot of these bands. Each of these bands is supposed to represent at least one protein, one polypeptide of that size. Problem is, we look right here, and that band, how many different proteins are there that are going to be roughly that size? We don't know. Is there a chance that there's multiple different proteins that are close to the same size? Sure. Yeah, maybe there's multiple proteins in this, whatever this cell is, that all have roughly 400 amino acids in them. But they're completely different proteins with completely different roles, different shapes, but they have about 400 amino acids, so they're going to be about the same molecular weight. That's the problem with this. You can't just sit there and look at that band and say, there you go, let me just cut out that one band, everything's pure. No. You take the soluble extract, you just busted open the cell. Here's all the proteins that are there. You go through those different steps from the previous slide. And notice here, there's some extraneous bands you could just make out. So other proteins came along. But as you go across here and you do all of those different things, you now get to this point right here where, well, we're pretty much going to say purified. You don't see any other bands present. That's how you go through and you do this. How far things are going to move is based on, as you see here above my head, quite a few other, variables. What's the charge, the velocity, electrical potential of the polypeptide, what's the overall net charge, friction coefficient of how it's going to be able to move through whatever, polyacrylamide gel you chose, or agarose gel, or anything like that. One of the things you will notice, though, is when you go and you read the literature, you'll hear this referred to not as a, just as electrophoresis, because there's so many different types of electrophoresis. But you're going to see it referred to as, SDS, polyacrylamide gel electrophoresis. Polyacrylamide's the type of agarose gel electrophoresis. You're putting it in you're putting your sample into an electrical field and you're trying to see how things move, distances and all of that. That first part though, the SDS, sodium dodecyl sulfate. Sodium dodecyl sulfate is a detergent. This is what we're going to use to give an overall charge to our proteins. We're going to get the proteins to denature. It allows them to relax their overall shape, their conformation, gets things to start to break, not to break down, but to become more linear. So that's not just, how compact the protein is or the polypeptide is, but its overall size is now going to be the variable we're measuring. Gives an overall, as I said, an overall charge. So that way it goes from negative to the positive. And it allows for things to separate out by weight, not compactness, not by charge. There is other ways of separating out based on how compact, and other ways to separate out by overall charge. Is this an overall more positive protein? Is it an overall more negative protein? SDS gets rid of that variable. So you can go through and you could go back to that gel and you start at the top and you load stuff into the gel and you run it down and then you stain it some way. Yes, here we're talking Comassi blue, but there are other stains out there that have an affinity for proteins. What you see here, though, in column 1, lane 1, is what is referred to as a protein ladder. A protein ladder is a man-made composition of different proteins, as you see here: myosin, beta-galactosidase, bovine serum albumin, ovalbumin, blah, 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 blah. We know the overall molecular weight of these. So we can run this out, and here it kind of forms a quasi-ladder. Yes, those ladders you climb on. That's where they got the name for it. So we know up here at the top, this first one is going to be myosin, 200,000. Down here, this one is going to be ovalbumin, 45. Down here, the smallest one that's going to move the furthest, lysozyme at 14.4 thousand. Okay. We can measure this distance. We can measure that distance. And as you notice above my head, from this figure in the textbook, you can graph that out. Along the y-axis, size. Along the x-axis, how far did they move? And you get this relatively straight line. Is it going to be perfect? No. Okay? Because there's going to be vagrancies in how the polyacrylamide cell solidified. There's going to be slight differences in buffers and things like that. Just, so it's not going to be a perfect straight line, but it's going to be close. So you put your best fit line, boom. Over here in 2, lane 2, you have your unknown. You don't know the size of this unknown. Same time you're running this ladder, you have this unknown, and it will run alongside. All of these will settle out. They will travel a certain distance in a given amount of time. This unknown will travel its distance in that same given amount of time. Well, just as you did with the ladder, you're going to measure the distance. Boom. Distance moved on the Y over to the X, our best fit line, and then down will tell you the overall size of this protein. And a lot of times students want to sit there and look and just say, hey, look, here's the protein. I'm just going to go over here. It's between 45 and 31, about halfway through. So that's 9. That's, 14. So I'm going to split it in half, and there you go. No, you can't do that. That's if everything is working linearly. Everything's moving in a linear boom, boom, boom. Though here everything's moving logarithmically. Okay? It's not a linear, this distance is the same between this and this and this. Because look, from here to here, that's 7,000. Just a little bit more distance, that's 10. About the same distance, we've now 14. This is 20-some, 30-some, again, 20-some, and then this is almost 100,000. This distance here is not 5 times here. It's a logarithmic scale. So you have to do this graphing. Now, we have developed methods where we use the isoelectric point of the protein as a mechanism for separating things out. Instead of running down a gel based on size, here we have just gel. Okay, it's going to allow things to move freely, and we have a field set up. Negative on one end, positive on the other, and things are going to settle out where they want to in this pH gradient of a gel. They're going to move because of the electrical field, but they're going to settle out. That gel we're running them through, that strip, has different pHs, a different gradient running through it. High pH on one end, low pH on the other. And things are just going to settle out where their isoelectric point is. We could go through and we could cut this up. Here's things that have an isoelectric point somewhere around, 7.8. Here's things, the isoelectric point around 5, things like that. Or we can take the things that have separated out based on isoelectric point, and then we can separate them out in another dimension. They refer to this as two-dimensional electrophoresis. The previous couple slides when we looked at gel electrophoresis, everything is moving in one direction. Okay, one direction down or sideways if you're talking to isoelectric. It's moving. It only has one plane it can move in, one dimensional. Here, you're going to separate out horizontally by isoelectric point. You're going to take that strip and then you're going to put it on top of one of those SDS page gels, but a much bigger one. So you've separated out left to right based on isoelectric point. And now you're going to run just your normal gel electrophoresis we've talked about, where things are now, big things are going to stay at the top, small things are going to be down at the bottom. So they've separated out left and right at our first step, our first dimension, in one apparatus, transferring to another apparatus, that whole strip, and now they're going to run from the top down to the bottom. Big things stay up near the top, small things down at the bottom. So what that's done is things, down here, small and have an isoelectric point that's up near 9. Up here, all of these spots Rather large proteins, isoelectric point close to 9. Over here, large proteins, their isoelectric point is going to be down towards the pH 3 range. Down over here, same thing. Small proteins, isoelectric there. So, and then you see each of these dots is a different protein. Because remember I told you with the vertical, we're separating an SDS page, and you get those bands, but the problem is those bands, that's just size. Are all of those things the same size? How many things are going to have 400 amino acids? Well, if there's more than one protein that has 400 amino acids, they're going to be relatively the same molecular weight, close enough to it. They're going to be, instead of separate bands, they're going to come looking at like they're one solid band. Here's the thing. If they have different amino acids, even if they're the same, 400, which means they're going to run, the same point across right there. They have different amino acids that they're going to have different isoelectric points. So our first one's going to be right there, and our second one's going to be right there. In the one dimension, they were the same band, but because we did 2 dimensions, their isoelectric points are going to be different, and they're going to be 2 completely different spots on our big gel here. That's kind of cool. Now, again, we try not to say, oh, look, we have this purified protein from this crude extract, And the way we're looking at this is we're measuring activity. Our enzyme of choice, how often is it there? Is it, how much of this substrate is being turned into product, I should say, okay? In our minds, we're logically saying more substrate to product means more of that enzyme has to be there, thus activity. Our specific activity is out of the total amount of protein there. Our enzyme is one of the thousands, so our activity is going to be here. But if we somehow enrich, so that instead of thousands, it's now dozens, maybe a handful of proteins, and our enzyme of choice is the more predominant. Remember from that table, specific activity shoots up 1,000-fold. So we have fewer proteins present, fewer amount of protein present, but our enzyme of choice is now the predominant one. And that's really what we're looking for.